Welcome everyone. Welcome to week number 3. We are into our term now. So let's see what we've got going on. So last week we covered pH and buffers and their importance to organisms. pH is a measure of how acidic or basic an aqueous solution is. And we know now that organisms love a nice neutral pH for the most part. Including ourselves. And buffers are chemical systems that help maintain that pH in a state of homeostasis, which is extremely important. Can't remember if we cover that in this term, but if you don't now, you probably will. Homeostasis is a balancing system, and that's what buffers do. They balance the pH to maintain it at a certain level. And this is important because organisms can only function at certain pH levels. This week we're going to learn all about cells. And so first of all, we're going to look at the two principles of diffusion and osmosis. Diffusion is where molecules spread out. So this is more chemistry. Molecules spread out from areas of high concentration to lower concentration until equilibrium is reached. And you understand this principle. If you were to squirt a squirt of perfume in the doorway of a room, you know that if somebody on the opposite side of the room won't smell that perfume, at least for a while, until those perfume molecules get evenly distributed throughout the room. So you understand diffusion. Osmosis takes diffusion a step further. So osmosis is movement of water across a semi-permeable membrane until equilibrium is reached. So our cells, all cells, are surrounded by membranes, and membranes are permeable to water. This is important. We have to have water in our cells to function, but we also cannot have too much water. So that's where osmosis comes in. I'll tell you a funny story here. I had a senior microbiology student working with me one time, super smart girl named Heather. And she was helping me with some of my research at Clemson. And she was doing some counts on some white blood cells. And she was sitting at the microscope right back behind me. And I heard her grumbling a little bit. And then she said, "Janie, what in the world is happening? I counted the number of white blood cells on this one slide on one side, and there were 4. And then I counted on the next side and there weren't any. And then when I went back and looked at the first slide, there weren't any there anymore. It's like they disappeared." I said, "Heather, did you put the cells in water?" She said, "Yeah." I said, "Heather, do you remember osmosis?" And she went, "Oh, no." Yeah, so she put the cells in pure water, and that pure water caused the white blood cells to expand and expand and expand because the water was moving into the cells through osmosis until those poor little membranes couldn't take it anymore and the cells just burst open. So that's what happened, osmosis. You're going to take a look at that in one of the labs. You're also going to look at different types of cells. So there are 2 basic types of cells in the world. There are bacteria, which are also called prokaryotes, before a nucleus. That's what prokaryote means. And so that tells you that bacteria don't even have nuclei, much less other organelles inside the cells. They are very simple cells, very small cells. Of course, they can cause all kinds of problems for us, but they also help to maintain the health of the whole biosphere. So bacteria are terribly important for the health of the planet. And so bacteria versus eukaryotic cells. So what are the eukaryotes? Those are plants, animals, fungi, and protists. So a protist is like an amoeba, that's an example, or algae, or are types of eukaryotes. Fungi, you know, is mushrooms, molds, those sorts of things. Plants, you know, and animals, you know, and we, of course, are animals. So our cells are very different from bacterial cells. You will be drawing examples of bacteria and eukaryotes and also viruses. Well, why didn't I mention viruses as a cell? Because viruses aren't cells. As microbiologists, we do not consider viruses living because they do nothing on their own. They are just little pieces of genetic information that get inside of host cells, take over like pirates, and cause that cell to do whatever that virus wants it to do, including make new viral particles. So I'll talk in just a minute about COVID and SARS-CoV-2 so that you get an idea of one of the viruses that is in the news right now. You will add structures to your cells in your viruses, and then you will identify them using a dichotomous key, which I will mention again in just a minute. So here's a picture of osmosis and how it works. If you look on the left-hand side of the screen, you see that there are different solutions that are separated by a semi-permeable membrane. The solution on the left has many more water molecules than it has solute molecules. A solute is something that's dissolved. So the solute molecules are in the water. There are few solutes and a lot of water molecules. On the right-hand side of that beaker, you have a lot of solute molecules, which means that you have fewer water molecules. So then look over to the right. What happened in this case? Water molecules from the left side, where they were in higher concentration, moved to the right side until equilibrium was reached for all of the water molecules. So you will see that with your dialysis tubing when you do that experiment. If there's more water outside the tubing than inside, the water will move in. If there's more water in the tubing than outside the tubing, the water is going to move out of the tubing. If the water concentration is the same, you will get no net change. All right, so let's talk about SARS-CoV-2 and COVID-19 because, of course, this has been in the news since at least March in our country. The coronavirus, as you all have heard of it being called, coronavirus just tells you it's one of a group of viruses. The more specific name is SARS-CoV-2, the virus that is making its way through the whole United States, it looks like at this point. So what does SARS-CoV-2 stand for? SARS stands for Severe Acute Respiratory Syndrome, and isn't that what this disease is? This is the name, and SARS-CoV-1 is actually a different coronavirus that hit, and I want to say back in 2009 in different spots around the world, but there certainly wasn't a pandemic of it. Right now we are experiencing SARS-CoV-2. CoV stands for coronavirus, and it's called number 2 because it is the second coronavirus of this type. COVID-19 describes the disease process. So CO stands for corona, V stands for virus, the VI stands for virus, and D stands for disease. So that's telling you this is the disease process. The shortness of breath, the tiredness, the coughing, so many other symptoms now. Inability to breathe. There's so many symptoms now. So that's the difference. One is talking about the virus itself, and here's a picture of the virus. And if you look at it, you see that it's got all these nice little spikes sticking off of it, which are important because those spikes allow it to stick to host cells. It is surrounded by an envelope, and I give you this in the description of the virus there. The envelope is a piece of your cellular membrane that it picks up on the way out of your cells. Now this is important because these envelopes are fairly easy to strip off of a virus. So some people say, well, the only way to get rid of the virus is to wash your hands with soap and water. Washing your hands with soap and water is great. That's my preferred method for cleaning my hands. But alcohol-based hand sanitizers also work very well. This is an envelope virus, which means that it contains phospholipids, fats, and fats dissolve with alcohol. So hand sanitizers work very well to destroy these viruses. It's an RNA virus, Some viruses are DNA viruses, but this is an RNA virus. And it is non-segmented. It has one strand of RNA, which, you know, that doesn't mean much to you, I know, but this is one way that we classify them. And it is a positive strand, which means that it can make its own viral proteins right away once it gets in your cells. And then, of course, I mentioned those spikes for attachment to the host cells. I've given you a website here. Just go to the CDC website, Centers for Disease Control and Prevention, and you can get lots more information about COVID and COVID-19 and SARS-CoV-2. Okay, well, what about the labs this week? It will take you time to do these 2 labs. So I put it at the beginning of this, time. You can always look in your lab to see how much time it is expected for you to take to do these experiments. Now, I will say that it takes me as long as those directions say. So I am guessing that it takes you a little bit longer than that. So just be aware of that. The potato experiment is the diffusion lab because you're trying to see how diffusion works with different sizes of potato cubes with iodine. So potassium iodide, which is your solution in this lab, reacts with starches to turn to a blue-black color. Normally it's a brownish, light brownish color. And when iodine or iodine compounds touch starch, they turn blue-black. So that's how you can tell if the iodine has diffused into your potato cubes. So this whole experiment is about diffusion of iodine into the potato cubes. The second part of the lab is the osmosis lab, which has to do with the dialysis tubing. This special type of tubing simulates semi-permeable membranes like our cell membranes. And so you will see what happens when you put different sucrose or sugar solutions in the tubing versus outside the tubing. I've listed things that you need for this lab. You're going to have to get your own potato, and you need a calculator, timer, a paper. Make sure you wear your safety gear, especially for the potato part of this lab, because iodine is very corrosive. Do not mess around with that. I know that you put it on your tissues, but that's a diluted form, so don't mess around with this iodine. I also mentioned that it's going to take you time, but there is 1 hour of downtime in the middle of these experiments. So you can go off and do something else during that time, but then you need to come back and finish up the lab. On page 24 of your lab instructions, in the last column, here's your lab report, but page 24, the last column, it says hypotonic, hypertonic, isotonic. And if you would just write in there under that, outside the tubing compared to inside the tubing, this will make more sense. So these terms, hypertonic, isotonic, and hypotonic, refer to a comparison between 2 solutions. So the example that I gave you before, where there were very few solute molecules but a lot of water molecules on the left, that is a hypotonic. The solution on the right was hypertonic. There were many more solute molecules than water molecules. It's hypertonic to the solution on the left. The one on the left is hypotonic to the one on the right. Well, how do you remember which one's which? Okay, hyper. What makes you hyper? Lots of sugar. So if you have lots of solutes, you have very little water. This will help you remember the terms. All right, and then you will draw out your 2 types of cells and your virus according to the instructions in the lab. And then you also need to include the identification of the bacteria that you've drawn, the bacterium, and the eukaryotic cell and the virus. So you have to use something called a dichotomous key, which is included in the lab. And this is how you identify. So, for example, with the viruses, at the beginning it says, is your virus a DNA virus or an RNA virus? So you choose which direction to go. And then you follow the instructions. It may say, if you choose the RNA, then skip down to step number 4. and follow that. Is it double-stranded? Is it single-stranded? Keep following the directions, and eventually you get to an identification. Dichotomous keys are used all the time in biology, all the time. This is how we identify organisms. A couple of other things. The experiment next week, for lab week 4, is a little bit long, and you have to get it started by Thursday in order to have any results by Sunday. So if you want to get started earlier than that, feel free to do that. You will be germinating some peas. Okay, and they're very specific instructions about what to do for the first few days while you get your peas germinating. It is terribly important to use non-chlorinated water. So distilled water, bottled water, not water straight out of the tap. You actually can use water straight out of the tap if you put it into a bowl and let it sit out for at least 24 hours. The chlorine will evaporate from it, but you have to use non-chlorinated water or else you will kill your peas. So I'm just saying, be thinking ahead, looking ahead to that lab to figure out what you need to do and in what time frame. I want to mention something else because I'm seeing some things in the labs that I'm grading. You all must be using the lab report forms, and I've given you instructions about how to find these. Under Content, Learning Modules. Start here, Assignment Guidelines and Rubric, and then Lab Reports. That's where they're found. It is a zip file. You have to download it and then open it up to get to your lab report forms. You've got to use these forms because all of the graphing information is there, all of the questions that you need to answer. If you just do the lab, you're missing a tremendous amount of information that you're graded on. So use the lab report forms. I'm also seeing that people are putting in lots and lots of different documents. Okay, everything needs to be included in the one lab report form. Everything. Every picture, every drawing, every graph, whatever needs to be included in that one lab report. Okay? And then finally, you're also going to start your final project. You go to Ask a Biologist website and you figure out which research question to select. There are 4 of them that are listed in this week's learning module. There are 4 labs that are listed, the one with the ice, the one with the nerves, the Manduka caterpillars. Oh, there's another one. Oh, air pollution. So those are ones that you can select. If you want to do something else, then you have to contact me ahead of time, and I have to approve it before you move on and do milestone number 1. So milestone number 1 is your final project, but it's all of your background information. So this is where you do your research. And here are 2 questions that I want you to think about and answer while you're doing your background checks. Why is this important in the grand scheme of things? Why in the world is it important to know how many pokes you feel with a paper clip on your arm as compared to your back? Why in the world is that important? You may think, that's the silliest thing I ever heard of. Well, you're going to have to figure out why is that important to know. And you're going to have to explain that. And also, what is known about this phenomenon? So you're going to have to do some background research and figure out information about this. If you're looking at the brine pockets in ice, why in the world is that important? What has that got to do with anything? These brine pockets, saltwater pockets of water inside ice. So what? But they are tremendously important, and that has everything to do with the environment. So find out what is known about these phenomena. And as always, if you need my help, just ask for it. I am here to help you. So let me know how I can help you. If we need to do a video chat, if we need to talk on the phone, whatever it is, if you just want to email me back and forth, whatever you need, I'm here for you and will gladly help you. Okay, I hope you have a great week. There's a lot to do, so let me know if you need my help.